By now, we know that the living organisms are classified into five main kingdoms. The first is Kingdom Monera, about which we talked in the last lecture. It mainly includes prokaryotic organisms, which are bacteria. Second is Protista, in which you have single cellular organisms. In the current lecture, we are talking about fungi. Fungi has a unique kingdom of life which is separate from plants or animals or bacteria. In this, the organisms have a complex and organized cells with well-defined nucleus as well as other organelles. Let's quickly overview the structure of fungus. Fungus has cell wall which is made up of chitin that is protective as well as supportive. They have plasma membrane which encloses the cell and regulates the movement of nutrients and waste in and out of the cell. Fungi form these thread-like structures called hyphae, which are the basic unit of their growth. Collectively, the hyphae are called mycelium. The cross walls between the hyphae, which divide the individual cells, are called septum. This septum also have some pores for some material to be exchanged between different cells. At the cellular level, the fungal cells have nucleus because they are prokaryotes, and fungus can also be multinucleated. They also have mitochondria, which is responsible for generating ATP. They have vacuoles in which they store nutrients and waste, and they also help in maintaining the shape of the cell. The fungi also produces spores, and the spores are inside this body called the fruiting body. Fungi can have different kinds of hyphae. First kind is the septate hyphae. In septate hyphae, there are cross walls dividing the hyphae into individual cells, each with one or more nuclei. Septa allows the cytoplasm and organelles to pass from one cell to another. The example of this kind of structure is seen in Ascomycota or in Basidiomycota. Xenocytic or aseptate hyphae is a condition where the septa is not present in the hyphae and there is a continuous flow of material as well as organelles between the different parts of a fungi. This kind of hyphae is found in lower fungi, which allows the rapid growth and flow of nutrients. Example, Zygomycota. Sometimes there are specialized hyphae present in the fungi. These specialized hyphae can be Hostoria, which are mainly related with the absorption of nutrients by penetrating into the host cell. Or they can be orboscules, which are highly branched structures that aid in the nutrient exchange in the mycorrhizal fungi. The nutrition in fungi is mainly heterotrophic because the fungi cannot produce their own food through photosynthesis and they have to rely on other sources for this nutrient requirement. They can be saprophytic based on whether they can decompose and absorb nutrients from the dead organic material around them or not. So example is mushroom that breaks down the leaf litter and obtains nutrient from that. The other mode of nutrition is parasitic, in which the fungi extracts nutrient from a living host and end up causing some disease in the host. For example, cordyceps infecting insects or rust infecting the plants. The third kind of method of obtaining nutrients is mutualistic. This is a symbiotic relationship between the fungi and its partner organism. A mutualistic relationship is one in which both the partners are benefited. For example, mycorrhiza, in which the fungi is interacting with a plant, or a lichen in which a fungi is interacting with an alga. Asexual reproduction in fungi is more common because it leads to rapid growth of the fungi under favorable conditions. The different methods for asexual reproduction are fragmentation, in which the hyphae just breaks and each fragment grows into an individual organism. Or the other method can be budding, in which a unicellular fungi, for example yeast, buds off from the parent cell and detaches and then becomes a new organism. Or the third method can be spore formations, in which the spores are produced by mitosis. These spores disperse and germinate when there are favorable conditions for their growth. The example is Conidia in Ascomycota. Asexual spores are produced in large numbers, they are genetically identical to their parent, and they are beneficial because they can colonize very fast. Asexual reproduction helps the fungi in spreading faster and helps in a short-term survival. Sexual reproduction mainly helps with genetic diversity and it aids in adapting to the new environment. Sexual reproduction in fungi can be plasmogamy, karyogamy, or through meiosis. In plasmogamy, the cytoplasm of two mating type fungi fuses, leading to the formation of dikaryotic stage. Dikaryotic means having two nucleus. Karyogamy is the fusion of nuclei to form a diploid zygote. So the next stage to plasmogamy would be karyogamy. And meiosis then leads to the halving of chromosome numbers, so the diploid end up becoming haploid. So we can see the alteration of generation in fungi. That means there are certain stages of life cycle which have haploid genome and certain stages which have diploid genome. But there is also a unique stage of the life cycle of fungi which is dikaryotic. That means there are two nuclei present in a cell. So the main body of fungi is primarily haploid. So haploid is the dominant stage of their life cycle, with each cell having only one set of chromosomes. Then for a very short amount of time they have a dikaryotic phase in which they have two nuclei. These two nuclei fuse to form the diploid stage, and then diploid stage undergo meiosis to form the haploid structure again. Let's quickly review the classification of fungi. Fungi are mainly categorized into two categories based on whether the sexual reproduction is present or absent. The group of fungi that does not have sexual reproduction is called fungi imperfecti or deuteriomycetes. Now in the category of fungi that have sexual reproduction, they can be primitive fungi or advanced fungi depending on whether their mycelium is aseptate or septate. The categories under primitive fungi are Phycomycetes and Zygomycetes, and those under the advanced fungi are Ascomycetes and Basidiomycetes. Zygomycetes are primarily terrestrial fungi present in soil, decaying matter, and they are sometimes parasitic. Their hyphae are cenocytic, that means they do not have septa separating these hyphae, which allows for continuous flow of cytoplasmic material. Zygomycota can reproduce through asexual reproduction or sexual reproduction. In asexual reproduction, the fungi produces spores through the process of mitosis. And in sexual reproduction, there is the formation of zygospores, which are resistant structures that undergo meiosis and then germinate. The common examples of this category of fungi are the common mold that we see on the bread, or mucor, which are present in soil and food and can also cause the spoilage of food. Next group of fungi are Ascomycota. They are also called the sac fungi because of the spore formation in a sac-like structure called asci. These are the largest fungal phylum that includes yeast, mold, and fungi like murals and truffles. There is septate hypha present in these fungi, which leads to the compartmentalization. Ascomycota also undergoes sexual or asexual reproduction. During asexual reproduction, they form conidia as the asexual spores, and in sexual reproduction, ascospores form in the asci within the fruiting body like the mural. The mating of haploid hyphae leads to the formation of dikaryotic phase that undergoes meiosis to produce the diverse ascospores. Common examples of this category of fungi are Saccharomyces cerevisiae, which is the yeast that we use in research, baking, and production of alcohol; Penicillium, which is the source of penicillin; and Aspergillus, which is used in fermentation, and some species of this fungi are pathogens of plants and human beings. Next phylum is Basidiomycota. They are also called club fungi due to the basidia that looks like a club. This group includes mushrooms, puffballs, shell fungi, and rust. In this fungi, the septate hyphae with clamp connection to the main dikaryotic cells are present. This kind of fungi can also undergo asexual and sexual reproduction, although asexual reproduction is rare with occasional conidia formation. Sexual reproduction is a prolonged phase in Basidiomycota in which the hyphae fuses to form dikaryotic phase. The basidia produces basidiospores in the fruiting body called the basidiocarp. Basidia undergoes karyogamy and meiosis to form 4 haploid basidiospores. Common examples of Basidiomycota fungi are edible mushrooms, the agaricus; amanita, which are the species of toxic or hallucinogenic fungi; Puccinia, that causes rust in several crops; and Boletus, which are edible mushrooms with tree symbiotic species. Fungi form symbiotic relationships with many different organisms. A symbiotic relationship is the one in which both the partners are benefited. The first kind of relationship is mycorrhiza, in which the fungi is associated with the roots of higher plants. The fungi enhances plant nutrition and water uptake, and plants provide carbohydrates to fungi. The other kind of interaction is lichen, which is the association of fungi with alga or cyanobacteria. The fungi offers structure and support, whereas the algal partner provides food via the process of photosynthesis. Lichens can thrive in extreme environmental conditions, and they are important indicators of air quality. Endophytes are the condition where fungi lives within the plant tissue without harming them. Often, this leads to increase of plants' resistance to stress, drought, and pathogens. There are several economic benefits of fungi. Let's have a quick review of those. Fungi is widely used in the food industry for the production of bread, beer, wine, and cheese, and there are several kinds of edible mushrooms available. In pharmaceuticals, fungi are used for the production of antibiotics. A common example is penicillin, which is obtained from Penicillium. Fungi are also used for the production of immunosuppressants and cholesterol-lowering drugs. In agriculture, mycorrhizal fungi is helpful in improving crop growth and soil health. Fungal biofertilizers reduce the dependency on chemical fertilizers in agriculture. In biotech, enzymes from fungi are used as detergents, biofuels, and in food processing. In the bioremediation, certain fungi are used for breaking down the pollutants in the environment, thereby aiding in the ecosystem cleanup. Let's review the potential of fungi as a pathogen. Pathogen is an organism that can cause diseases. So, fungi can cause diseases in plants. They cause diseases like rust, smut, or blight. For example, Puccinia causes the blight in wheat. This leads to significant agricultural losses and affects food security. In animals and human beings, the fungi is able to cause several diseases. For example, a superficial infection causes skin infection like athlete's foot or ringworm. Fungus can also cause systemic infections which severely affect immunocompromised individuals. For example, candidiasis caused by Candida and aspergillosis caused by Aspergillus. Fungal diseases are a major threat to the biodiversity that affects wildlife population. Losses in agriculture, healthcare costs, and efforts in controlling fungal pathogen has severely impacted the economy. To summarize, fungi are eukaryotic organisms which mainly depends on other sources for obtaining the nutrients as they cannot perform their own photosynthesis. They are characterized by the presence of hyphae, their cell wall has chitin, and they can undergo both sexual and asexual reproduction. They are essentially the decomposers of the ecosystem where they clean up the dead and decayed material through the saprophytic action. They are also associated with several other organisms in a symbiotic relationship, and they are also pathogens of plants and animals. They have valuable use in the food industry, pharmaceutical industry, agriculture, biotech, and in bioremediation. And they cause several diseases in plants and some important diseases in animals and human beings as well.